This is the United Podcast Network.
This is Voices in Validation, brought to you by IVT Network. IVT Network is your expert source for life science regulatory knowledge. Voices in Validation brings you the best in validation and compliance topics. We interview industry experts from pharma, biotech, med devices, and laboratories. Here is the host of Voices in Validation, Stacey.
Thank you, and welcome to another episode of Voices in Validation, brought to you by the IVT Network. Each week, we bring you timely interviews with industry experts from pharma, biotech, medical devices, and life science manufacturing, with discussions focused on top trending topics, frequently asked questions, and industry best practices. This week, we are pleased to have with us Brian Nunnally. He's the Global Head of Quality Control at Securus. Welcome, Brian. Great to have you.
Thank you so much, Stacey. I'm so glad to be a part of this series.
Yes, it's excellent. And we are talking this week about managing quality control and variability in the life science industries. And for our listeners, we've seen many advances in technologies and guidances in the pharmaceutical industry over the past decade. But one thing that hasn't changed is the expectation that pharmaceutical manufacturers are required to monitor their processes to capture unexpected variabilities over the life cycle of the product. So before we leap into this subject, Brian, can you give us an overview of current guidance on the subject?
Sure. And again, thanks for inviting me. I appreciate it. I love these kind of opportunities to talk about this kind of thing. It's a real passion of mine. I go back to the process validation guidance that FDA issued in 2012. And it seems like 8 years ago, but it seems so modern and fresh today. I think one of the things that kind of replaced the idea of this kind of N equals 3, right? We do this process validation, we do 3 batches, and all of a sudden everything magically works from there on. I don't think any of us ever really believed that that was really the case. It really is about understanding our data a lot more than that. Sure. We need to have a broader perspective and understand, hey, it's about really process understanding, right? So this is all validation. It's not a discrete exercise. It now becomes, method development. I'm going to be very analytical specific in this case. Method development is part of method validation. You have the validation exercise, and then you have all the work after that where you're still learning and you're still developing information. And I think FDA really recognized that they wanted that to be the case. They didn't want us to have this aspect where we go through, we do this one little tiny exercise, and then we create this wonderful little document that we shunt away in a corner and gathers dust until we need to bring it up because of an inspection or something like that. And I think it really is meant to be replacing the knowledge with this kind of continuous aspect that we're constantly learning, and we're applying those learnings back into the process. And so, in my own experience, I've seen a lot of times where we've seen, investigations or things that didn't happen and say, wow, this is interesting. We didn't know that was the case, or we didn't know this was the source of variability. And we've had to adjust accordingly for it. And it's been very powerful for us. So I think that's the guidance. If I was going to point to someone and say, boy, you really need to understand one piece of guidance, that's the one I would go to.
Thank you, Brian. And that's so important and valuable information for our listeners, because I think a lot of us in the industry get hung up on the guidances as, we don't want to get a warning letter. We don't want to, you know what I mean? Yeah. So we're always trying to manage that we sometimes forget that we need to really manage the quality of the product at the end, that we're going to put out there. And if you're doing all that you can to produce the highest quality product throughout the entire life cycle, all of those other things are automatically going to fall into place, it seems, right?
Absolutely. And you're really building that quality in. And I think, FDA and other authorities, health authorities, recognize that you build quality in from the ground up. And that what you're getting at the end is merely just a test of what the process is going to do, or the analytical method is going to do. And so I think that we get caught up a lot of times in looking and thinking only about things in that context, instead of really thinking about it, like you said, that we're really looking that guidances are really not telling us maybe what requirements or anything, but telling us the right way to do things. And really the process validation guide is a perfect example. That was something the industry basically had been talking to FDA about it, and FDA is coming about and saying, yeah, we agree with you. So from that aspect, I thought it was really nice.
Absolutely. Absolutely. Thank you. So you mentioned being analytical, and obviously, I mean, analysis that starts by looking at historical data trends, right? So because you mentioned like the company who said, hey, we didn't even know this was a source of variability. But if you're watching the data trends and really doing your best analysis, you should reveal those weaknesses well before you get to the end of the process, right? In any of your methods. So, because past performance is the best way to predict future performance, I would say. So, this is important in understanding and reducing and controlling variation. And what are some of the current control variation tools and tactics that you've seen or use currently in the pharmaceutical industry?
Yeah, I think I'm going to tell you. Let me tell you a story. I find it best. I'm a storyteller, right?
I love stories.
I think it's the best way to communicate things, and so I'll tell you a story. So it comes from my past. I had a production manager come to me and say, "Brian, what's wrong with your laboratory?" And threw some data in front of me. And I remember it distinctly. It's like I can remember like yesterday. I'm seeing the person. I'm seeing all of this, from this aspect. And I remember looking at the control chart that they were demonstrating. It had a few points on it. It wasn't a very big control chart or anything like that. But what I remember about it was the 2 points they were talking about were really high. And it was a case where if you looked at those two points in particular, they were above. It was a yield spec is what they were looking at, showing a yield chart. And it was above 100%. And so I knew that it wasn't right.
Right.
Everybody knows you can't get a greater than 100% yield.
Exactly.
Something's wrong somewhere. And, I will say a cautionary note for those process folks that have tuned out because they think I'm talking only analytical. Let's remember process variability. And I know how some of the process people measure. I remember we used to measure in one of the companies I was at, they measured volume with a stick. so yeah,
not the most efficient.
Not the most accurate way to do that. So I know those volume measurements cannot always be accurate too. So it's important to remember that. But anyways, I knew this was a problem with the laboratory measurement that was being made. It was obvious to me but the thing was, a lot of the folks came and said, "Hey, the problem isn't accuracy. You're not giving us accurate numbers." And accuracy and precision are two things to understand, and they're two. You need to understand which is which in each case, because if you use different tools to attack the different problems. And so I recognized that this problem was a precision one. It was very clear to me that the method had been validated, and the precision was about 15% was the validation percentage, and I suspect it was much higher than that. And we actually went through. And we were working on variability reduction already. So this was a perfect event that I needed, the burning platform to make something happen. So I went back and we looked at the data and we said, look, we know this is not an accuracy problem. It's just the precision's too high. They need about 2% variability and we're giving them about 15.
Okay.
That's not going to work.
Right.
And so we said, what can we do? And so I had a manager at the time, really, really bright guy. And he sat down and said, what we need to do is we need to get the analysts involved with this. So we got the analysts involved and we gave each one of them a method. The reason we gave each one a method was we wanted everyone to be able to own something and to make the decisions for everyone else. So if they didn't get their way with one other method, they got to win at least once if they wanted. And they were awesome. They did great. They took a hold of the methods and they harmonized everything. They harmonized how long they set the sample sit out, what pipetting they used for the assay, how many times they were inverting to mix. They did everything. And we had done a DOE, a design of experiments, right before this event happened. And we had looked at the variability that we had associated with it. And we re-executed that design of experiments after this process was done. And for that assay, we saw the controllable variability for the analyst controllable variability fall by 50%.
Wow.
Immediate. It was immediate. There's no change in technology or anything like that. We have the variability right there. What's interesting was one of the other materials we used, the variability didn't decrease at all. And we were just astounded by that. We went back and we realized that there was a dilution step. And the way they were doing the dilution step, they weren't controlling for that. They were just kind of basically doing it once. And there wasn't any replication. So we realized if we replicated across that dilution step, we would take down the variability by about 75%, was where most of the variability was coming from.
Wow.
And so, I mean, there's an example. And again, I tell people this and they're astounded. We had another method that we saw the variability go from about 10% RSD down to 2%. That was overnight. It was an overnight change. And what was really exciting, and I tell people all the time, is it's not difficult. It seems hard. The hardest part about this example that I used there is deciding to do it. It's deciding that I'm going to do it, and we're going to make this a priority, and we're going to execute against this.
Right.
And when you get that kind of management fortitude, I'll call it, you really kind of can transition things and get a lot better. And I tell you what, it was one of those things. I saw the production manager that did that some years later. I was visiting one of the other sites, and I happened to run into him in the hallways. And he looked at me and he said, "Brian, you really made your bones with us with that investigation."
Wow.
And so it really comes back to I talk about in my bio that being the best quality control unit is my passion. And I don't believe you can be the best quality control unit unless you are focused on reducing variability and delivering better data to your customers. And like I said, it can be a complex process, but I've found in my experience that the most complex part of it is deciding to do it and actually making it happen.
And so that was just looking at control charts and design of experiments, and then you figured it out that
it was really literally just easy. A matter of just the analysts and them getting together and saying, you know what, we're going to have one way of doing it, and we're not going to tolerate doing it any other way.
Right.
And it's funny that it seems so hard yet so simple in that respect. And so I think and one of the questions that people always ask is, well, how do you institute that into the system? And I tell them the best way to do it is you have your validated ranges. So it'll say incubate for 4 to 6 hours, and you don't want to write a deviation because someone incubates for 4 hours when you're standard. Everybody's going to do it at 5.
Right.
So what I often tell people is, fine, leave 4 to 6 in there and put target 5. So that it's very clear to everybody in training and everything what you're supposed to target. But if someone incubates for 5.2 hours, you're not writing a deviation that you're going to say is not a problem.
Right. No, I think that's so important to have those exact specifications outlined so that people everywhere, because a lot of times, these larger pharmaceutical companies, they have multiple locations where they may be producing things, and it's obviously necessary to make sure that everyone globally is kind of following the same process.
Absolutely. And I think that's a major concern, is that you share that information across there. And there certainly is an attitude of it wasn't invented here, right? So you're going to always have that kind of issue when you go between labs. And so the more local you can make this kind of effort, the more successful you're going to be.
Sure.
But certainly share those learnings, share that knowledge across the network whenever you can.
Right. And that's very important. So are these tools that we're talking about applicable to other industries, say medical device as well? And can you give us examples from your past, more examples, because that was a great story, of how you have implemented some of these tools?
Yeah, I think one of the funny things is these tools are readily applicable. And I would love to say that they're new, but many of them are over 100 years old. When you hear about Duran and Schuhart and Deming and all these people, the gods of these, statistical kind of things, that I listen to. John McConnell is my mentor and taught me all of this, and he learned from Deming. So I consider myself a grandchild of Deming.
There you go.
From that aspect. But, I think these things are readily applicable, and you see them in every industry that mining, petroleum, optical lenses, the lenses you're wearing right now, or probably had something to do GE, Six Sigma. It's all the same thing, and it all applies. And so what I found is when I've talked about this at conferences, that I find that I get all kinds of people come up. I get process people come up and say, well, can I do this in my area? I'm like, sure. I'll tell you a great story. One of my favorite stories I remember, and John McConnell told me this story. He was doing a session one day, and he was talking about a situation where they had to they had to go and they had to, they had a fermentation process. So here's a process example, right, for all you process people listening. They had a fermentation process, and it was real important that you get that number right, okay?
Right.
Fermentation broths are notoriously heterogeneous. So you have to really mix well to make sure you get a good sample, and you determine that sample, and that sample would determine what you did in the next part of the process. So it's very important you get that number right, because if you overcharge the next step, you would create yield problems. If you undercharge, then you wasted your time, and time is money, right? So we didn't want to do that. And so went through, and he was describing the process, and one of the operators in the front started to look very agitated, started to even cry a little bit. And he was describing the process. So the process was, they were supposed to go through and they would take and they would agitate the tank, and then they would stop the agitation. They would take a sample, and then they would agitate again. And then they would take another sample, then they would agitate again, and then they would take another sample. So you ended up with 3 samples that you delivered to the lab. The lab would take and take each one of those samples and do 1 run on each HPLC. Excuse me. And so then you have 3 numbers. You would then combine together to give you 1 good number that would average across that variability. Heterogeneous samples, more samples are better to average across that variability. So he was sitting there, and he saw this agitated operator, and he stopped the thing and went and said, "What's wrong?" He said, "Well, the way I was trained to do it was to agitate, to take a sample, and then go 1, 2, 3. So who gets the blame in this? The lab, of course the lab gets the blame, because the lab would go and show, we've got very tight RSD. Those numbers are very close together. Why? I mean, we're giving you the best number possible. But of course, we know the problem was all in the sampling, and the sampling variability was not being considered. And so that's an example of how, making sure that everybody's doing it the same way. And if you have a training regimen where you say, this is what you want done, you need to have some audit to make sure everybody's doing it that way. And also, it probably talks about the lessons, and we'll not talk about it today, of training someone that trains someone that trains someone that trains someone. The evolution of training like that. How you don't want to see
the copy of the copy of the copy.
Exactly, right? You lose resolution with every time. So,
exactly.
No, I think it's real important that we think about things like that. So that's, I think, another good story of indication how it can be used everywhere. And I can tell you, everywhere I've gone where I've talked about what we're doing from an analytical laboratory perspective and how we're addressing variability, I always have those process production managers who, first of all, say, thank you for doing that. Thank you for giving me better numbers. I appreciate that. And do you think it would work with my groups too? And I'm happy to tell them, yes, it will.
Yes, absolutely. And thank you for, shining a light on the need for training, because I think that
Absolutely.
So often across the whole entire life cycle, no matter what department you sit in, I think you can only be as good as the training you receive.
Absolutely. 100%.
Yeah. And I think it's interesting that you're noting the tools that you use are 100 years old. Let's just round it off.
Yeah.
And yet we have so many emerging technologies that we are now introducing into our processes and into our systems, or integrating with our systems. I'm curious, have you seen a significant change in the process controls being used, or the way that data is analyzed as we, get more digitized, or, bring in some AI or, Internet of Things? What are you seeing there?
The funny thing is, it's the new tech is really interesting. I think it's helping us get a better handle on the information. And, it's funny, Deming was really against computers, or people presumed he was against computers. He wasn't. He wasn't against computers at all. He loved computers and saw the advent and the way that they would be used in the future, probably better than most of persons of his time. But I think what's interesting about that, if you really go back to it, Stacey, is that there's some value in just plotting points and getting a piece of paper and some graph paper and plotting the points out. And like I said, I was trained by McConnell, and when I do my training as well, I force people to do it. And it's not that, again, I love computers. I use computers on every aspect and technology. I got like 2 cell phones and all this kind of stuff. I use lots of technology. Right. And I love technology. But I find that you have to learn kind of how to do it right the first time and really see the data that way. The problem with computers that come in is they give us the opportunity to kind of ignore some of the data and to and have a chance to really not get to work and massage and really play with the data like we want to. Now, let's be honest, with all the work that we've got and everything that we've got, we have to use computers. And I find that there's a lot of technology out there, a lot of things that are very useful in helping to create control charts and to create metrics and help us to better be able to see the information that we have. So from that perspective, I'll always say I'm a big proponent of technology and love its use. And I think we found with computerization and digitization and the big data advent, there's a lot more opportunity to get data that we could collect that in the past we just didn't have an opportunity to deal with. And so
Sure.
I think you have the ability now to take all this information together and to do some really excellent work around correlations and start pulling this data together and see things that maybe aren't easily observed unless you have a computer to really crunch the numbers and to see the data. So it's really kind of important, I think, that you kind of think about that, aspect. And I think we're going to see two things I would say as a cautionary note. One is a cautionary note that remember that not everybody is in the same place as you might be from a statistical perspective.
Sure.
Probably some of our listeners right now are listening to me talk and are clicking off and saying, oh my gosh, he's talking stats. I don't want to hear anything about that. I don't believe any of that. 80% of all stats are lies, or something like that, and I think that it's important to remember not everybody's in the same place. So
Right.
You need to, as we get to data and want to be more data oriented, it's important to remember to gauge where your audience is and bring them along. If they're not where you are from a data perspective, don't browbeat them with the data. Bring them along. Help them to understand where you got to the point that you got to.
Sure.
And the other thing to remember is, I think back to an old story I heard where I walked into a room, an office, and there was a control chart on the wall. And this was another McConnell story. He looked at the control chart and he looked at his buddy and he said, "What is that control chart? It's really interesting. I can see all the trends and everything like that." I said, "Yeah, every time I take my break and kind of sip my coffee, I look out the window and I count the number of blue cars that I see, and then I put a little check mark by it." So not everything that can be measured needs to be measured,
Sure.
nor does it need to be analyzed. So it's important that while we can gather a lot of data, it's important to really understand, what do we really want to measure? What's really telling us something? Be careful of those campfire stories of people that tell you things that don't matter that really do. But keep in mind, there are some things out in this world that really just don't matter.
Absolutely. So it sounds like, as we are integrating more technology and that, obviously people still need to have the understanding of how the data got to be, where it is. Like, you get the numbers, you do run the analysis using the computer, let's say, but you still need to understand what the data is saying, whether that's possible, right? I mean, because, you're the checks and balances, let's say. But it seems also like you're saying that it may be making it more efficient.
Absolutely.
Quicker, right? Like you can run numbers faster, and you still need to understand the process, but I mean, there's a lot more numbers that you can process in a shorter period of time, I would think.
Absolutely. That's exactly the case. And I think we've got the ability to do complex mathematical analyses that we couldn't do, 20 years ago.
Sure.
And now are readily available and easy to do. Any iPhone can do it, kind of thing. So I think that's the power in the analysis now, that our tools are so much better. It's kind of like going from the Bronze Age into the Gilded Age now that we're in. And I think we've got so much further to go. I think the opportunity to collect more data and collect more information and to really utilize that in some powerful statistical analyses, we are only scratching the surface on right now. And I think that's where the future's going.
I would agree, Brian. Definitely. I think we're just at the beginning of this. People across the industry are looking at small changes and small integrations to start. I mean, some of the larger companies are much further along, of course, but a lot of the companies are just sort of trying to figure out how they can automate their processes, let's say, and do other, smaller things. So we're still right in the beginning stages. I can't wait to see, how changed we are in a decade or two, for sure. So Brian, thinking about the life cycle approach, quality controls are necessary throughout, like we've already mentioned. Can you talk about this as it impacts each area of the pharmaceutical or medical device process, from R&D straight through post-approval changes?
Yeah, I think one of the things that a lot of times you hear about is, R&D, we don't have enough sample size, or we can't do enough N to really make variability reduction a valuable thing. I disagree completely. In fact, where you really want an understanding of statistics and all the tools that I've talked about is in R&D.
Mm-hmm.
And one of the things that we did in Securus, for instance, was creation of what I call an AS&T, Analytical Sciences and Technology. And part of that analytical science and technology is not to just help out my QC group and make sure that they're redeveloping assays or developing new methods for existing products, as well as solving problems within the quality control unit. But it's also to go back to R&D and to work with R&D to help give the QC perspective to our R&D colleagues. And they do a great job. That's one of the things where we have at Securus, a lot of cooperation and a great group of folks that are working together very well. And so I'm real proud of the work that they're doing. But I think that R&D effort is so important because everything you do there translates directly back to manufacturing. So the more you understand the sources of variation back further, the better. I can think back to a story I heard from someone in another company, talked about how they were having problems with an assay, and they said, "I don't understand. We were having problems. It was still in R&D at the time." And they're going, "I don't understand why we're still having problems." And they went back to talk to R&D scientists. Well, they said, "Oh yeah, we saw that before." So, well, it's not in the development history report. What are you talking about? It's like, well, we didn't document the ones that failed. You got to document that. You got to talk about when the method fails. So I think it's real important that I don't use any names in here because I don't want to the guilty to know their guilt, but I think it's important that we think about that documentation of the failures and making sure we're looking at those sources of variation. And then when you get it into quality control or you get into the process and you're now actually manufacturing, it's important to kind of capture all that sources of variation, what you learn from that. Deviations can teach you a lot.
I think the failures, you learn from the failures way more than you learn from the successes. And that's always been the case, right?
And I think one of the things is even look at those things that are out of expectation. Don't even look at the ones that are out of spec. Look at the ones that are out of expectation. And if you're really doing good control charting, you can really learn a lot from those out of expectation results. I think a lot of times we tend to look at things only the in-spec, out of spec kind of thing, rather than really looking at very robustly and think that some things can be still in-spec but worthy of an investigation because they give us something different and they may be giving us a signal. I think some of those times those differences can even be good. I mean, you can envision where you had a very high yield for something and say, hey, why did that happen? We would like to replicate that.
Yes,
we can get more material out of the same thing we're doing right now. So that would be a great thing. So I think it's important that we kind of look at all of those aspects and not just consider the OOSs and things. And I think then when you go further down the process and post licensure and even into times where you start tech transfer to other more mature sites or things like that, tech transfer is a great opportunity to share that learning and document what you've got so far. And you get new sources of variability, right? When you start talking about reproducibility, going from one lab to another lab, right. You have a great opportunity to learn more about that method. And then, as you talked about, now you're sharing methods across different sites. It's really important to go through and share those sources of variability and make sure you built into systems ways to knowledge share between the places running and doing the same things. So the thing you want to avoid most of all is you don't want to learn something twice.
Okay.
You can do that. It takes work. It's not easy. It takes work. And I think that's one of the things that some of the things we've done here at Securus and having the global quality control under one group, one person, really has helped to kind of drive some of that learning. So I think that's an important thing to not forget.
Of course. I mean, and talk a little bit more about the controlling variability. Maybe once you've released the product and it's out there, because we hear about change control a lot post,
yeah,
right. But What
Yeah.
control do you have over variability at that point?
And I think that's one of the things that people kind of miss as well. And I'm glad you brought that up, is because you think about change control as a perfect example. Is when you hear this harmonization and everything that I preach here at variability reduction, sounds like I don't want to hear, you don't want to have any changes.
Right.
Far from it. I love changes. I love improvements. I want them. I beg for them.
Right.
From an efficiency perspective, what I want everybody to change together. So I want us to say and find new ways of doing things, improve ways of doing things, and then all of us move in concert together. And I think if we can do that, that's where things get really powerful. And I think change control is a vehicle to make sure that all the affected parties can really be brought into the discussion, and making sure that we're changing in a way that everybody's aware of the change that we're making, so we stay harmonized and we move together. So I think that's where some of these tools can be used very, very effectively.
Thank you. Yes, and I think that's very important. So thinking of that concept of everybody changing together, being kind of uniform in that effort, and as we already talked about with the onset of Industry 4.0, and we're getting more digitized and more automated, and the idea is that we achieve higher levels of quality and compliance. Because manual or human errors are decreased. And that's assuming that everybody's working as a unit, right? And variabilities hopefully then are less as a result of just that process. Do you have a sense of the impact on testing times and agility? And how about, ROI, QC costs, as we start to reduce variability that comes from human error, let's say?
My goodness gracious. That's the thing that people, you always ask me, why do you do this? Why is it important for variability? And I tell people, it's very easy. Think about just about any quality metric or any metric you would use that would talk about good performance. Variability reduction will help you in just about every one.
Right.
And there's a concept called Little's Law, and Little's, it's not Little's good idea, it's not Little's really good notion. It's a law. It says that if you can reduce the if you reduce variability, it's a kind of a corollary to it. But if you reduce variability, you will see increases in throughput. You will see, because you kind of have to.
Mm-hmm.
The equation is set up such that if you reduce variability, you'll see less work in progress. You'll see lower amount, you'll see higher throughput, or some combination of the two. It has to happen. It's just based on the equation. You can't do anything but. And so what I found in my experience is when you see full implementation of reduced variability, you see things like your invalid rate. So for instance, in very QC specific, you see your invalid rate go down, your deviation rate goes down, your OOS rate goes down, your turnaround time goes up or goes down as well. You're able to get more out in the same amount of time. You're able to do more work with the same amount of people. You think about it, it's intuitively obvious. If I have less invalids, if I have less deviations, it means I'm not having to redo work as much. So that means those people that were normally redoing work are now able to do new work.
Right.
So it's intuitively obvious that all these things are going to happen. And if you can do more work with the same amount of people, you're going to save money. If nothing else, you're going to be able to do more work with your fixed cost. And you can look at some of your variable costs can be decreased as well, because now you're not consuming extra reagents and things like that you have to do. So any way you look at this, you're gonna save money and make things better. And my experience as well is that if you decrease variability, you will get more grams out at the end of the day. So if you're a process person, if you want to get more grams out of the same space, the same people that you've got, variability decrease is the way to go, and really focus on that.
Well, when you put it like that, it seems so obvious.
And you would think it was, Stacey, but I tell you what, it's still something I find that when I say it to people, I get a lot of responses, all kinds of things from people that just either they don't believe it or they don't accept it, and it won't work for us, and there's always a reason behind it. But I tell you, it can work for anybody.
Yeah, no, and it does sound like, everything's just as efficient as it can be. Less bumps in the road makes it a much straighter path, right?
Absolutely.
So,
Absolutely.
So it definitely does make sense. And while automation solutions, so you talked about ROI, obviously goes up,
absolutely.
as the variability goes down, and while automation solutions may have the potential to deliver strong ROIs, the industry overall is still struggling to capture the true value in implementation of these technologies and these processes. What do you see as the most common drawbacks? What's holding people back, or industries, companies back, from really buying into this whole notion?
Yeah, it's really interesting, Stacey, because I've seen some my experience 20 plus years in this industry. I've seen a lot of automation implemented, and I've seen a lot of places where it was discussed and everybody got really excited about it and then started choking on the cost of implementing the actual automation solutions.
Right.
And it's interesting, I think, because and computer system validation didn't help. When we started bringing in Part 11 and everything like that, everybody started to kind of freak out a little bit about what that's going to mean for automation solutions. So I think we're kind of still recovering from that, haymaker, if you will, of what that meant. Part 11 is great. I mean, the idea behind it and making sure that that the computer systems are doing what they're supposed to do. Right. It makes perfect sense. I'm not here to bash it. Don't get me wrong, but I do think it's important that kind of created, I think, some of the situation that we're struggling with automation. But the other factor is, frankly, it's just cost. It's a lot of investment right off the bat, and at the end of the day, you look back and say, what are you getting out of it? What is your return on investment you talked about? If you're not willing to say, I'm going to take a human being that's working and I'm going to remove that human being from the workforce, it's hard to generate some of the return on investment that you're going to need to be able to really sell that in a very tightly competitive capital environment. And so one of the things that I found as well is that a lot of times automation doesn't really remove people from the equation. It changes the equation. It changes the work that is being done. So instead of seeing that we put these automation in robots and now we can get rid of 3 analysts, that's not how it usually works.
Right.
What happens is you're now changing the work that the analysts are doing. So what I find is it really enables you to be able to do more with the people that you've got. So oftentimes you can expand your laboratory operations, or you can do more tests, you can support more businesses, do more things that the company's been asking for and wanting to do that you just couldn't do before. But the other real advantage of it, and this is where the ROI can come in, is you can do things better.
Right.
Because robots can do some things much better than humans can do. And I think I'll give an example is dilution steps. Those are, rife with repetitive injuries. And so people, notorious, have the carpal tunnel and other kinds of things that are very difficult. And you do repetitive stress of pipetting is a real thing. Right. And so I think if you can get a robot, that can come in and do those diluters, an auto diluter or something like that, that can save you variability, certainly, but it also can save you those stress, injuries, and safety issues that you're going to have. It can be ergonomically a very good thing to do. So I think we need to broaden our definition of what we want for automation and not look strictly at, is this going to let us replace people? No, let's look at it more broadly than that. Is it going to help us get better data? Is it going to help us with less safety incidents? Is it going to help us, ultimately get turnaround time decreased for a variety of different reasons? All those have a cost associated with them. They're harder to quantify, but a lot of times automation can really help us with those kind of things. And I think we need to be more broad in our definition how we go down this road.
Right. I think that's a great point. And it goes right back to what we were saying at the very beginning of our conversation with, if you're always striving to have the highest quality, to be the best that you can be as an organization, to produce the best products, and to your point, to treat your employees, the best, then you're going to end up in the place you want to be at the end. Absolutely. So, and hopefully automation is part of that. I think it is. I think automation is absolutely going to be part of that. It's easier to do, and I think it's easier to do if you can get it in the R&D environment so it can translate into the quality environment. But it's even harder, I think, to justify automation in the R&D space than it is in manufacturing. And so with that in mind, knowing that at some point in time, I think everyone in the pharma and medical device industry is going to fully make the shift to automated processes and other integrated technologies. how, in your mind, can pharma and device companies discover which of the innovative tools and technologies will have the greatest impact, immediate impact on their business, and then maybe the long term? Because I think if what you're saying is true, that the biggest hurdle is the money and the time that it takes to see the return on that investment, then perhaps if there's small steps they can take or things they can do right off the bat to improve things a little bit and then work towards a greater solution, that might be, that might be the catalyst for getting some people over the hurdle.
I think so. And I think I always say that one of the things that I think partnership, it really comes back to partnership and collaboration. I've been very fortunate in my career that I've had some great partnerships and collaborators in the support industries, I guess I would call them. And that's our partners that are vendors that are out there that are selling equipment. And I've found in my experience that I can always find someone that's a little on the edge that wants to either say, hey, look, bring your samples in. We'll get a material transfer agreement. Let's bring those samples in. Let's do some running in our lab. Let us show you what this can do. Or they'll say, look, you know what? I'm gonna find you a piece of equipment. I shouldn't do this, but I'm gonna find you a piece of equipment. I'll let you have it for a little while. You show how it works, and then I'll just trust that if it works, you'll want to buy it. And it's funny how that works. That kind of little things like that can make a big difference. And you can make a big difference when it comes down to that justification for the capital committee that you're going to. If you can look at them and say, look, we've proven it. Here's the work we've done in our lab, or here's the work we've done in the process. Look at what we're seeing here. I recognize you can't do that with a tank, but you may not be able to do that with some very expensive pieces of analytical kit. But I can tell you, if you can find ways, if the vendors out there are listening to me, I think if you can find ways to get that equipment into the hands of the users, or you can take their samples and show them how you can do what you can do, I think you're going to find that sales is going to be a lot easier.
For sure. And I think, to your point, if the vendors really take the time to understand what the pain points are.
The ones that really good are the ones that really, the questions they ask aren't, let me tell you all about my equipment. They're asking questions. What do you need? What are the problems? What, if you could solve anything, what would you want solved? I mean, it's kind of silly questions like that ultimately I think are the benefit to us, because when someone asks me that, I can tell you the problems I need to solve. Let me start with, and start to work through that. And I find those vendors that are out there that can do that are the ones that I patronize for sure.
Right, I agree, and I think if they can match their solution to the biggest pain point for that organization, you're going to have a win. Right?
Yeah, for
sure. Brian, great conversation. I have loved your story so far, and we're nearing the end of our time. But before we go, I did want to give you an opportunity to share some last tips and or thoughts with our listeners on variability, but also, really working to get the highest quality that you can in your particular industry, department, organization, whatever.
The funniest thing that I find, one of the most common questions, Stacey, and I think the most important thing I want to leave folks with, a lot of people ask me, oh my gosh, I love what you're talking about, Brian. I would love to do that. I just have no idea where to start. And I tell them, you know what? It doesn't matter where you start, just that you do.
Great
and so,
point.
There's a lot of ways to do it. You can parse it out. You can say, what are the tests that manufacturing uses? If you're in an analytical lab, what are the tests manufacturing uses? And then figure out the ones where they use the number. They take your number and then they, do something with it. If that's the case, then you want to make sure you attack those numbers first, because anything that those measurements, those are the ones that are going to translate the variability directly in the process. If you're on the process side of things, then start early, because the more variability you have early in the process, the more it's going to kill you downstream.
It's only going to grow, right? I mean,
absolutely.
It's a small problem in the beginning. It's going to be a big problem in the end.
Absolutely. Little's Law will tell you that. So, from that perspective, start in the early part of the process, because that's where the transformation needs to happen. And I will promise you, if you commit to this, and I don't make many promises, but I will tell you this, if you commit to this and you really do it, and you get serious about it, and you get people aligned around this and get the management fortitude to make it happen, you will see these benefits, I promise. Lower variation, lower deviations, lower invalids, better numbers, better customer service, lower cost. I mean, all these things just go hand in hand. And I promise you, if you do it and you just commit to it, it will happen.
Absolutely. It all makes sense, Brian. It all makes sense. And listeners, hey, you have the promise right there.
That's right. You don't get many promises on these things, I would bet.
That's right. I don't get many promises. So there it is. Excellent. Well, thank you so much, Brian. I really do appreciate you taking time out of your day today to be with us. It's been great chatting with you. And also, before we leave, I want to give a big shout out to our producer, Ben Kitchen, and the Studio 21 Podcast Cafe. And lastly, a big thank you to you, all of our listeners. We do value that you spend your time with us listening and learning. If you have specific topic requests or guest suggestions, we'd love to hear from you on that. If you enjoyed this episode, we ask you to subscribe in your podcast player of choice and share with your friends, colleagues, and your online networks, because that helps everybody learn more and it helps us grow. And we do appreciate that. So for today's show notes and information on the podcast, please visit www.ivtnetwork.com. We'll be back again next week with another great interview. Until then, stay healthy and safe.
The views and opinions expressed by the hosts, guests, or callers of this program do not necessarily reflect the opinions of the Studio 21 Podcast Cafe, the United Podcast Network, its partners, or affiliates.